again, we are back. I'm sure some of you will take a minute to join us, but Rich, Ronnie and I just finished our last live stream, but we're going to answer some of your questions right now. So basically, I know some of you are asking us some biology, chemistry, and maths questions. Those of you will be new to the channel. So throughout this week, and if you buy the course and decide to continue using us, then you're going to have us basically until you finish your A-levels, but you can submit your questions to us and we're here to help you out. So I know that thanks for those of you who just joined us in that live session. It was super good, and lots of interactivity on the chat, which is what we like to see. Something that we're really into at TT is like effective learning practices. And so being interactive and being active in what you do is going to really help you. Sitting there sort of learning and twiddling your thumbs and like not really paying attention to what you're doing is going to make basically studying very ineffective. So by interacting and joining us live, you are basically going to increase your chances of what you're committing to memory. Am I watching the Man United derby match? I kind of stopped following football. I don't know which Rich you're talking to, but yeah, I'm not a huge football follower anymore. I vaguely support Crystal Palace, but not really in any. I couldn't name half the teams to be honest with you anymore.
Yeah, I'm not a football fan either. It's not my bag. So if I'm watching anything sneakily off the side of the screen, well, I'm not watching anything. But if I was, then it probably wouldn't be football. I'm not sure what it would be, but there we go.
I would sneakily be watching Richard Feynman's lectures.
Oh, yeah.
That is what I would sneakily watch. Yeah, for sure.
It's a bit geeky, Ronnie.
It's not that it's a bit geeky, yeah, but he's amazing. He's such an amazing teacher for someone that intelligent and with that amount of knowledge that he can communicate ideas that are beyond 99.9% of the population in a way that makes you feel like you understand and makes you feel like you are learning from. He's brilliant. I seriously recommend Feynman lectures for anybody at all, regardless of whether you think you like physics or not.
Hi Jemima, hi Elizabeth, hi Warder, hi Noor, hi Ryan, hi Akid, hi Sugar. So yeah, the Feynman technique, exactly. So did we have any questions? I just didn't want to, I wanted to end that last web sort of broadcast with just a nice clean instructions for anyone who hadn't used us before. Was anyone confused by anything that we just did there? I know we had a little bit of confusion about the 1 week versus the 1 month. Any other things that you'd like us to clear up? If you've got any, we'll do one biology, one chemistry, or maths question. We'll try and pick something. Hi, Sarah. Two gifts. Ryan and Sarah.
I think the questions that I know that people are worried about are coming up a lot in maths, but we can do this on Thursday unless anyone's got anything specific, is hypothesis testing and mechanics, force diagrams, resolving forces, that kind of thing. So I'm happy to do on Thursday, which is maths day, I'm happy to do some hypothesis testing, maybe correlation hypothesis testing or binomial distribution hypothesis testing, and some mechanics. But if anybody's got any specific questions, then shout. Akid, Rich, this is such a good question. Do you guys do a video on revision techniques and research? Oh, yes.
100%, 100%, yes. So like
everything we've done, everything we've made is based on a huge amount of research. We're really interested in effective learning techniques and trying to make it basically like we were actually, Rich, chemistry Rich, were you lazy at school? Like me and Rich were?
I was very lazy at school. I was so lazy.
Yeah,
I took
exactly.
years to do my A-level.
Yeah, exactly. So basically our whole approach to education right from being teenagers has been, how can I get the best possible mark with the least possible work? And we bring that philosophy to this as well. Like we're about making it as effective as possible. So if you're going to spend an hour with us, then we are going to make sure you get the absolute most you can possibly get in terms of learning from that hour. So everything that we do is based on that, but particularly when it comes to sort of March, April, May, then we're much more focused obviously on exams and exam technique and effective revision strategies and using Pomodoros, using the FAST technique, all those things. Yeah,
so 100%. There's lots of 100%. That was exactly my motivation. I figured out basically it was taking me longer to achieve learning stuff and finishing projects when I was not paying them full attention. I would be, Facebook was basically launched when I was at university. Facebook was becoming a big thing. in 2004 to 2007. I had my first Facebook account in 2004. That's when my Facebook account goes back to. And like, in my first year, it didn't really matter. I didn't do that much work and I didn't need to. And then eventually I figured out that this is getting important now. I'm paying £50,000 to go to university. I may as well actually come out with a decent grade. So I wanted to figure out, how do I get out the most from the time that I put in? It's not rocket science. It is a bit of brain science, but not rocket science. Basically, no distraction. You need to give your brain 100% focus on the thing that you're doing, because any distractions, any division to your attention, this is the reason I don't have a floating head in my videos. You know, the YouTube lives we have, the little minimized view, that's not ideal for learning, because you're looking at me or are you looking at the screen? Are you looking at me or are you looking at the screen? And as soon as I split your attention, your ability to remember either of those things, it's not half, it's way less than half, because the brain doesn't know what to focus on. So huge amount of learning technique goes into all of them.
Yeah, Hussein was just saying, actually.
Yeah, I love it. Hussein's question.
Can we explain the order and how to use the TT? So does he watch the video first? Does he make notes at the same time? I mean, what we recommend, highly recommend, is you sit there and you watch it twice. So you sit there, you watch the video from start to finish, you focus on it, you take it all in, and then you go. Hopefully you print off those skeleton notes that we've got, those on the. You pause it, you write it down, you make sure you understand it. You watch the next section, you pause it, you write it down. So first time, from start to finish. Second time, that's where you take your notes, pausing it in between. Don't let it run whilst you're writing, because whilst you're writing, you're not listening to what's going on the video. So twice is the way forward. So it sounds crazy, like you're watching the whole thing twice, but it's much more effective than sitting there reading a chapter of a textbook twice, for example.
Oh, 100%. You're not trying to join those dots up. You know, if you're trying to write and write, if you're trying to listen and understand and listen and write notes, they're two tasks that's going on right there. And again, you can't quite do both at the same time. So what you need to do is watch it all the way through, get an understanding, get the overview, get the understanding in first, and then second watch. Most importantly, if you're writing, the video is paused. Because if you're writing and the video is still going, you're then getting a bit behind, and then you're trying to catch up, and then you're splitting your attention again. It all comes back to focus and attention. So 100%, that's what you do. And don't forget to use the traffic light system. Don't forget to ask comments and questions, all that kind of stuff. So yeah, definitely.
Yeah, I mean, like one of the things that Rich and I did this summer was that we took one of each other's videos and we did precisely that, just to see how it felt. So watch it through. Then make the notes, pausing it when you're making notes. I got in trouble because I didn't pause it when I was making notes. I just tried to write really fast, because I do write really fast. Yeah, I know, I got told off. But then we talked to each other about what we'd learned from the video. So I asked Rich questions about the maths that he'd done. He asked me questions about the biology, just to see what had gone in and what hadn't gone in. So yeah, everything that we do, and we want feedback from you as well. You know, you watch a video and you think this one was really good, it was really concise, and I was able to really learn from it. Let us know. You watch a video and you think, what? Let us know, because we can redo it, we can think it differently. We want that feedback from you as well. We're not just giving you a resource and saying off you go. That's the whole point of the Ultimate Guide, is that we're working with you to help you with that learning.
Okay,
there was a chemistry question.
There was a chemistry question.
I'm all over it. I'm typing away.
Okay.
Oh, you're going to type
on
the
screen,
answer.
it's about
Write the following ionic equation between calcium chloride solution and sodium hydroxide solution to give a something.
Yeah, that's it. So essentially with ionic equations, all right, you only include the ions that are forming the precipitate. So you might have two complete salts. So that was sodium hydroxide and calcium chloride and sodium hydroxide. Well, you can ignore the chloride and you can ignore the sodium completely, because they just don't get involved. So your full equation, your full ionic equation to form that precipitate is just between Ca2 plus and the OH minus, because that makes the calcium hydroxide precipitate. So you need Ca2 plus 2 OH minus, and that gives you Ca brackets OH brackets 2. So Ca brackets OH2. And that'll give you, so ionic equation only include the ions that you've got in solution. I'll write the full equation out in the comments now.
Arjun said, can I have a can I ask a maths question? And I said yes, and I've waited and waited and it's not there, so I'm a bit disappointed. So I'm quite excited for that to appear in a minute. But the other comment that I wanted to point out is Seb says, Rich, I love your background. Your Heisenberg background.
Oh, absolutely.
It's amazing. And yeah, I need to.
I'm sitting around, I can't see it. Like,
it's so good. And I need to find like an equivalent mathsy one so I can put it behind me. Definitely. Oh, here's the maths question. Use the quotient rule. Yes. Right. I'm having this. It's maths time. Okay. Use the quotient rule to find the derivative of x squared over 1 minus root x. Okay. I'm going to have to grab a board. Okay. Because maths just talking sounds like nonsense. So if I do this, can you see the board? Is there enough light on it?
I think there'll be enough light, yeah.
Yeah. Okay. So, quotient rule. I actually cannot see myself, so I don't know what you. Hang on, let me change this so I can see what you can see. Let me try writing this, and then see if you can. Yeah, you can see the whole board. Cool. Okay, so the quotient rule is for differentiating a quotient, and what we do is we take the bottom is going to go on the bottom squared, and then on the top, this guy will go first, multiplied by this guy differentiated, and then a minus, and then this guy on his own, and then this guy differentiated. So all we need to know is what is the ingredients, and in this case it was Rich, can you check I've remembered this right? Blah blah blah, x squared over 1 minus root x.
Yeah.
No idea what you're talking about.
Yeah, x squared over 1 minus root x.
Okay.
Oh, against the question.
Yeah. Okay. Look, it's not complicated. Look, you have
x squared over 1 minus root x.
Right. Okay. So we've got u is x squared because we're doing x squared over 1 minus root x. So it's like plug and play. It's literally he is now x squared. So there is going to be the derivative of x squared, and there it's going to be x squared. This guy is 1 minus root x, which for the purposes of differentiation, it's a lot better to write it like that. So this v here is this guy. This v here is the derivative of this guy, which is minus a half x to the minus a half. Pull it down and take one off, which Rich said the other day was rude. Now I can't stop thinking about it being rude, even though it's been 15 years of me not thinking of it in that way. So thank you, Rich. And then on the bottom, it's going to be this guy squared. That's it. You just plug everything in the correct place. So the derivative is going to be x squared is the u. So here it will be x squared, and here it will be 2x. And then on the bottom, we've got 1 minus the square root of x. So this one is going to be minus x to the minus a half, and this one is going to be 1 minus root x, and then on the bottom you have 1 minus root x squared. And then there's maybe some little tidying up you can do on the top. Usually with the product rule or the quotient rule, there'll be a little bit of, like, for example, we've got a common factor of x. So you could pull out the common factor of x, and then you could deal with the fact that this is 1 over root x. So you could times the top and bottom of this by root x, but that's all kind of cosmetic. That is the derivative of that. Okay.
Awesome. Yeah, I think it is kind of like plug and play.
It is plug and play. It's really fun.
You just got to know which bits are which bits.
Yeah.
And then practice a little bit. There's a couple of questions. One was like, if you buy the year 13 courses or you subscribe to them, do you get year 12 content as well? Of course. Yeah, the courses are now linear. Year 13 students have to know all the year 12 content. We wouldn't produce a course that, you have to buy 2. That's dumb. So yeah, absolutely that is. There was a question there about chemistry. I can't remember. I haven't seen any biology questions yet. There's a few people asking what's the code for. Watch the YouTube live we've just done. The recording will be up pretty much now, I think. So you can go that. We went over all what the codes are about and how you can get it free for the week or for the month, depending on what you do. Okay, we've got, is there a way to work out what precipitate is given rather than simply memorizing all of them? Rich, that was a chemistry question that I saw. I still know biology questions.
Oh, right. Okay. Yeah. I mean, there's rules. I mean, there are rules for, let's say, formation of precipitates. They are solubility rules, essentially, and they're not that hard to remember. I just think, there's stuff like all group 1 salts are all soluble, so you'll never get a precipitate of anything with group 1 in it. all nitrates are soluble. So you'll never get a precipitate of something nitrate. So, there are other rules as well, like as you go down group two hydroxides get more soluble. So hydroxides are higher in terms of solubility going down the group. Sulfates are smaller, so solubility gets less as you go down the group. And then carbonates can't. They can't dissolve. So all group 2 carbonates are precipitates. So there's a whole bunch of rules. I think there's 6 main rules we'll see you through, and they're in the tutorial that I've done. So yeah, then you don't have to memorize them all. Just learn the 6 rules and you're done.
In terms of color?
In terms of color, there's just no 2 ways about it. You just need to remember them. 9 times out of 10, they're white precipitates.
That's what I was going to say. If in doubt, white precipitate.
Yeah, 100%. Is that what Hussein's just said?
No, it's what I remember from my chemistry.
Oh yeah, absolutely. If in doubt, white precipitate. It's always your iodide and your bromide. They're slightly off white, but if it's going to be colored, it's going to contain a transition metal. So it's going to be like iron or copper or chromium or something like that. But all the others, white. Done.
Okay, I've got a question, a ballsy one finally. I'm just actually, I'll write in the live. Okay, so the question is, in anaerobic respiration, what is the final electron acceptor? I nearly misinterpreted your question then, because it's actually a bit of it's not possible. Okay, so the final electron acceptor during aerobic respiration, you do glycolysis, you do link reaction, you do Krebs cycle, you're building up all these things, and you're doing all of that so you can sort of charge up the batteries a little bit like the photosynthesis we talked about in the YouTube live yesterday. And the electrons are then flowing through a membrane, which is then pumping the hydrogen ions across a membrane to build up that proton gradient. And it's exactly the same in photosynthesis as in respiration. But you need that electron transport chain to be working in order to have yesterday, when we looked at photosynthesis, we looked at the electron being excited, leaping out of photosystem 1, and then being mopped up by NADP. Now, in respiration, it doesn't get mopped up by NADP. That electron has got to go somewhere. That final electron acceptor, the words tell us what it does. And that final electron acceptor, Ronnie, you can even as a non-biology person, respiration?
Yeah, breathing.
Breathing. There is actually respiration is the reactions. Breathing is the sucking air in and out your lungs. So there is a bit of a difference there. But like in that, what are some of the products of respiration? If I breathe on the
carbon dioxide.
And one more.
Carbon dioxide.
What's going to be on the mug?
Water vapor.
Water. Okay, so water.
Is that right?
Yeah, 100%.
I guessed that. Cool.
So the final electron acceptor in aerobic respiration is oxygen. Oxygen grabs all those protons, the hydrogen ions, and all the electrons. And so oxygen and hydrogen combine to make H2O to water. That's aerobic. But there is because in anaerobic respiration, you're not doing link reaction, you're not doing Krebs cycle, you're not doing oxidative phosphorylation, and therefore there is no electron transport chain, and therefore there is no final electron acceptor. So it's a bit of a false question. What happens there is that you basically convert the pyruvate, gets converted to lactate in animals, and it gets converted into ethanol and then ethanol in plants and yeasts, but don't worry about that. Basically, you convert it to other things, but there's no final electron acceptor. It's a specific key term that's reserved only for electron transport chain in oxidative phosphorylation. Lots of words there, but basically there isn't one because you don't do electron transport chain.
Rich.
Free coupon. It's now Try Ultimate Free. The Try It Free expired yesterday. Try Ultimate Free replaces it.
Rich, 2 questions. Can you explain codominance and multiple alleles? I just learned it today and it was a bit confusing with added blood groups. And then the other one, if you can hold 2 questions in your head, is why does the wave of contraction start at the base of the ventricles and spread upwards?
Okay, 2 pretty simple questions. Water, I'll just finish. Your teacher said it was pyruvate and ethanol. the term final electron acceptor is only used for electron transport chain in oxidative phosphorylation. So you should not be referring to ethanol or pyruvate or lactate as final electron acceptors. They may accept those electrons, but you don't call it that. It's not going to get you any marks in the exam. Maybe they were talking about in principle. Okay, so ventricles, why do they contract from the bottom up? Well, your tubes out the heart come out the top. If this is your heart, then the tubes poking out the aorta and pulmonary artery popping out the top. Now, the heart has got to squeeze from the bottom up so that it's like just squeezing a tube of toothpaste. You wouldn't squeeze a tube of toothpaste starting at the whole end and squeeze the other way. Doesn't make any sense. You want to squeeze from the opposite end, from the bottom end, squeeze all that stuff out so that the blood pumps up and goes either to the lungs or to the rest of the body and then starts the circulation. So that's why the ventricles contract from the bottom up. And the most likely question you're going to get on that is about the ABM delaying the impulse through the bundle of His, which causes the atria to empty before the ventricles contract, and then they contract from the bottom up. Okay, other question was about, testing my memory.
Codominance and multiple alleles.
Alleles. So alleles are variants of a gene. So let's pretend eye color is controlled by a few, it's actually polygenic. You can have a huge range of eye colors. But let's say you've either got blue, you've got brown, or you've got green. They're the only three options. So the blood group is like the AB, the AB, the ABO. So that's a better example. But let's go eye color. It's easier to understand. If you have alleles that are, one is dominant over the other. Let's say brown is dominant to blue. If you have two versions of each allele. One comes from your mum, one comes from your dad. If I've got one brown allele and one blue allele, my phenotype, what my eyes actually look like, will be brown, because brown is dominant over blue. Let's say blue and green are co-dominant. They are equally dominant as each other. Now, if I've got one blue and one green, my phenotype, my eye color, will be intermediate between, it'll be bluey green. It won't be blue, it won't be green, it'll be somewhere in the middle. So it's when two alleles, where you have to be heterozygous, which means you have two different alleles. The alleles are not the same. Blue and blue, you're gonna have blue. Brown and brown, you're gonna have brown. Green and green, you're gonna have green. If you have green and blue, you'll have a phenotype that's intermediate between the two different alleles. They are co-dominant, both, and the definition is that both traits are demonstrated in the phenotype. So there's an intermediate phenotype, basically.
I don't know whether this is biology or chemistry. Hardy-Weinberg?
Hardy-Weinberg's biology.
All yours, Rich, go.
Okay, Hardy-Weinberg is basically, there's a few assumptions. It assumes that there's no immigration of population. It's a way of basically seeing what's going to happen to the genes or the alleles in the population. Really hard to do without giving you some examples. But if I said 25% of the population have got the blue eye allele, and you only ever get Hardy-Weinberg questions with two alleles. You'll never get 3 or 4 alleles. It doesn't work, basically. So let's say there's just blue and brown for now. 25% of the population have blue eyes. Therefore, 75% of the other alleles in the population must be brown, because all of the alleles are either blue or they're brown in this example. And therefore, you can basically do some numerals to figure out how many have blue and blue, how many have blue and brown, how many have brown and brown. Join a web class on that or watch the video, because it's very difficult. Without doing worked examples on the board, it's hard to say.
Rich, we've gone biology crazy. Can you give a brief explanation on the nutrition work in tapeworm?
It's not in the spec, so not particularly, but yeah, so my knowledge is pretty broad. But so, I mean, what was the word after nutrition? The nutrition
work in a tapeworm? It might be a typo. That's what it says.
It doesn't mean it doesn't ring any immediate bells. But what they do is basically they've got a little, I think it's called a radula. I'm not 100% sure that. It's like basically little hooks that will hook into your small intestine.
That's disgusting.
So basically, if you imagine the small intestine as being like a river, obviously it's a slowly flowing river, but you've got foods moving through your alimentary canal. They hook in with these little hooks, and then the contents of your small intestine are being broken down by your enzymes, by bacterial enzymes, and there's all these nutrients floating around, and they basically just absorb the nutrients that are flowing past them through your gut. And then they will absorb it by simple diffusion. They have a high surface area to volume ratio, which allows them to do that.
Nice. That's lovely.
Hussein's talking about, sorry, this was a question, sorry, Nora, I forgot before to ask about, I swear we've talked about this before, but I can't remember, the EduQuas.
EduQuas is exactly the same as WJC.
Oh, okay.
Yeah, that's
so it. Is
So
WJC?
anyway, basically, yeah, it's WJC for English schools and colleges.
Okay.
Who's got that? Who's got that? No's probably one of 100 people.
But English colleges can do WJC, can't they?
They can, but they're now phasing out WJC in English schools and replacing it with the same. I'd say, I was going to say something rude then, but yeah, same thing, different name.
Yeah, that's all it is. Apart from it's linear, because we've still got AS and A2 in modular in Wales. So
Elizabeth said, can she ask a maths question? And I said yes, and then she didn't, so I'm a bit sad.
Okay, well, let's do another, we'll do till past. There's a million things happening today with lots of emails. Remember to share the course with your buddies this week or your classmates. It makes a huge difference to us. All we really want to do is show as many people the course as possible and let them try it out for free. There was a quick biology question there. I'll do my last one. Could you give a brief explanation of the body's defense against pathogens? Okay, so we have two major categories, non-specific and specific. The specific ones, let's say I get influenza, I get the flu virus, my body will go through produce a whole load of antibodies that will be specific to that specific strain of influenza. So that is a specific immune response. It's going to be your antibodies, your T cells and your B cells. I'll give you a very quick overview. The whole thing is going to be a bit of an information overload. You've got non-specific defenses against pathogens. Your skin, your pathogen just lands on you. Bacteria will be landing on you by the million per day, but they don't go into you because you've got skin, a physical barrier. You've got mucus and your nose hairs, and all these things are just going to prevent them getting into you. Obviously, you have a cut, your skin is broken, you can get a bacterial infection or a fungal infection can go into your skin. So that's non-specific defenses. Also, let's say a pathogen does get inside you. You have these things called phagocytes, which are generalist. It doesn't matter what kind of bacteria or pathogen you are, it's basically going to come and envelop it. And the word is engulf. And then it will fuse some enzymes with it and put it in a little sac and then release lysozymes in there, which will dissolve it. It will literally just munch up. And that's non-specific again. So they're phagocytosis. What it will do is present the antigens from the pathogen onto its surface, and that will trigger the T cells. And the T cells will then be like, the pathogen is here. Let's start making some antibodies to it. And there's a whole load it goes through clonal selection and then clonal expansion. That immunology section of the course is quite detailed. Check out the videos. There's maybe 6 videos that cover everything you need to know. In an hour, you could be the boss of immunology.
Elizabeth's maths question. Basically, the way you've typed it, Elizabeth, the expression 3 cos theta plus 2 sine theta 3 cos theta plus 2 sine theta is to be written in the form r cos theta minus alpha, r cos theta minus alpha. I mean, that in itself, r cos theta minus alpha, r cos theta minus alpha is r squared cos squared theta minus alpha. And you could write it like that, but I think you've typed it in wrong because it says where r is bigger than 0, r is bigger than 0, and alpha is between 0 and... The way you've typed it in, it doesn't particularly make sense. I could probably fiddle with it, but I think it's typed in wrong. So what I would suggest is join for free using Try Ultimate Free and just go on the trick section. In fact, the R cos X plus A video is here to be uploaded, but you can still type a comment in there. So if you put an upload of the photo of the actual question in that R cos X plus A section in the trigonometry, then I can reply to you typing, or I can upload a photo. That's probably the easiest way, because sometimes with maths, particularly something like that, where you're talking about several lines of manipulation of algebra and trigonometry, it's easier to do it by sending each other pictures than typing or talking. But the way you've typed it in does not make sense. So if that's what it says in your question, it's not answerable, because for a start, r cos theta minus alpha, r cos theta minus alpha is r squared cos squared theta minus alpha, apart from anything else. Apart from all the other things, apart from the fact that there isn't a common factor of r squared in that expression, etc., etc. So yeah, go on, sign in to Tailored Tutors, even if you just do it just for today, and go on Trig and go on r cos X plus A and send me a picture of the question and I'll respond. Did
Awesome. We're at 1:30. We've done a half an hour. We're back at 8 o'clock tomorrow. If anyone has got any questions for us, go check out the course, get involved, ask us questions on the course. I'm going to get around to doing as many comments as those today. I think I've had lots of comments on those going on the course today. I'm going to be on it as much as possible. Come back 8 o'clock, hit the bell so you get reminded of every time we release a new video on YouTube. And yeah, spread the word, share the love. See you all tomorrow.
See you guys.
See you tomorrow.